In this chapter, we're going to examine muscles, muscle tissue, and the different types. When you look at the two images I have here on the introductory slide, on the right side, you see an image of a man that has obviously been doing a lot of exercising, lifting weights, and developing these muscles over what you normally see on the average person. Now, if you look at the image on the left, you'll see that the skin has been removed. So we've taken off the superficial layers so you can actually see the position and the appearance of these muscles in the cadaver. Before we begin learning the individual muscles and more about the muscle tissue, we need to understand why the muscles are so important to the body. Well, the big thing is muscles cause movement. They move the bones that they're attached to and allow us to move around. We also use muscles for facial expressions. You can look at someone and see if they're happy or sad. But in addition to that, we have muscles that are important for vital functions. We breathe because of a muscle called the diaphragm. We use muscles to help us move our food around and to swallow it so that we can digest it and get the nutrients. And of course, we communicate by moving muscles associated with speech. Muscles help us to maintain our posture, stabilize our joints, and keep the body in its position by maintaining a state of partial contraction. Muscles also protect and support. So we package internal organs deep to muscles, and it helps to hold them in place. We regulate the elimination of waste products by using circular muscles called sphincters that control the passage of things at certain orifices in the body, such as the urethra that we use to the urethra has a sphincter that we use to expel urine, and the anal sphincter, of course, allows us to remove feces from the body. And another really big thing about muscle is that it helps us with thermoregulation. Every time muscles contract, they are breaking the bonds of ATP. Now, I know if you're in my class, you've heard ATP a million times. Well, remember, that is our body fuel. It takes fuel for everything to happen. You can't drive your car without fuel. You can't move your muscles without fuel. So contracting muscles uses a lot of ATP. So think about when you're cold. How does your body react to that to restore homeostasis? Didn't take you that long to think about shivering, did it? So when you shiver, that is muscle contraction. And muscle contraction breaks the bonds in ATP and releases energy in the form of heat. Now you know why you shiver. Skeletal muscle tissue is made up mostly of muscle cells, which have certain characteristics. Let's take a quick look at those. So excitability is the ability of a cell to respond to a stimulus, whether it's stretch or a chemical stimulus, that causes a localized change in the resting membrane potential. Now remember, you heard about resting membrane potential previously, so it's the same idea. You've got the potential for a cell to do a job by changing the difference in the electrical charge across that cell membrane. Now, once that happens, that electrical charge needs to be sent down the length of the entire cell membrane. So we call that propagating an action potential. The electrical signals function to connect the plasma membrane of the muscle cell to the inside of the muscle cell. So the stimulation actually happens outside on the cell membrane, and then inside the cell is where contraction will occur. So that takes us to our third characteristic, which is contractility. So there are contractile proteins found in muscle cells that will slide across each other, and that is what causes muscle to cause movement when those muscles shorten due to the sliding filaments. Extensibility is simply the ability to stretch. So those contractile proteins slide across each other and decrease how much they overlap, they stretch, and that is called extensibility. Now, Elasticity is the last characteristic that we'll look at just briefly. And elasticity, you know what elastic does, right? It stretches, extensibility, and then returns back to its normal length. That is the characteristic that is given by elasticity. Just like you learned when you were studying the skeletal system, each bone is an organ. In the muscular system, each muscle is an organ. So we have many different types of tissues that make up what we think of as a muscle. Obviously, you've got to have skeletal muscle fibers. There are connective tissues that separate and bind things together. There's an extensive network of blood vessels because we got to get a lot of oxygen to those muscles. Why do we need oxygen to get to muscles? Man, if you didn't say ATP by now, y'all might as well get me fired, because if you don't know you got to have ATP for muscles to contract and the blood carries the oxygen so we can make ATP, yeah, you knew that. We also have to have innervation. So with skeletal muscle, now remember, skeletal muscle is the muscle attached to the bones, skeleton, right? So with skeletal muscle, we have nerve cells that actually drop chemicals onto the cells to cause them to get excited, which means we're going to disturb that resting membrane potential and cause these muscles to contract. Muscle fibers are muscle cells. So if you look at this image, each one of these little bundles of things you see right here, those are muscle fibers. Again, a muscle fiber is a muscle cell. Okay? Now we're going to look at the smaller structures that you see here as we continue, but right now, each one of these little round bundles is a muscle fiber. Now, we wrap a bunch of those fibers up together to form this structure, which is a fascicle. So a fascicle is made up of a bunch of muscle fibers wrapped together with connective tissue coverings. And then, of course, the muscle itself is a bundle of fascicles. Fascicles are a bundle of cells, and a muscle is a bundle of fascicles. Now, as you've seen with other things that we've studied, we have connective tissue components as well. So these are layers of connective tissue that wrap around muscular structures. So the words are going to appear very similar to things you've heard. Epi always means upon. Remember, we saw the coverings of cartilage. We saw coverings on bone. Well, we've got coverings on these muscles as well. So epimysium, upon the muscle, we have the connective tissue wrap or sheet that covers it. So your epimysium covers the entire muscle. Your perimysium, peri means around. Paramysium is what you see illustrated by the white here. Paramysium wraps around your fascicles. This is also where your blood vessels and your nerves get sort of anchored in place. Endo, remember, endo means inside. Your endomysium is going to surround the muscle fibers themselves. So remember, each one of these little bundles right here is a muscle fiber. So endomysium wraps around the fiber. Paramysium wraps around the fascicles. Epimysium wraps around the entire muscle. Additional connective tissues associated with skeletal muscle include the things that attach muscles to something. So muscles can attach to bone or skin or other muscles. So the connective tissue that does that is called tendons or aponeuroses. A tendon is a cord-like structure made up of dense, regular connective tissue that attaches a muscle to a bone. An aponeurosis is the same tissue, but it is more of a flattened sheet. So if you look right here at this image of the dorsal aspect of the hand, you can see these cord-like structures. Those are tendons attaching the forearm muscles to the bony structures, okay? Now, when you look at the abdominal area, you see this white sheet of connective tissue. That is an aponeurosis. Specifically, that is called the abdominal aponeurosis. You have an epicranial aponeurosis. Anybody want to guess where that is? Epicranial, upon the cranium. So it's on the top of the head. So your aponeurosis are flat sheets that tend to attach muscle to other muscle or to skin. More commonly, you have tendons that attach them to bones. Now, your deep fascia is dense, irregular connective tissue that is superficial to the epimysium. So, superficial to, closer to the surface than the epimysium. Do you remember what epimysium was? It was on the last slide. The epimysium, remember, is the covering of connective tissue around the entire muscle. So then you would have the deep fascia that would be superficial to the epimysium. So it separates individual muscles or binds groups of muscles together that work together, such as your hamstrings. Your superficial fascia is made up of areolar and adipose connective tissues, and they are superficial to the deep fascia. So they are closest to the outside of the body, and this is what you see like with the subcutaneous layer that you learned about when we studied the skin. Muscle cells have the typical structure of a cell, but some modifications that make it have the ability to contract. So let's take a quick look at some of the microscopic anatomy. So the sarcoplasm is the cytoplasm of the cell. Sarc means flesh. So typically if you see something that says sarc, it is referring to something to do with muscle. So the sarcoplasm has the typical organelles plus additional proteins that function for contraction and some specializations of some of the other organelles. These skeletal muscle cells, now remember they're also called muscle fibers, they are multinucleated, so more than one nucleus per cell. They have a sarcolemma, which is the plasmalemma. Again, that's just the membrane. So the sarcolemma is the muscle cell membrane. It has voltage-gated ion channels that allow ions to move in and out of the cell across the membrane, and that, as you've seen before when we talked about depolarization, allows for the conduction of electrical signals. Now that sarcolemma, again, muscle cell membrane, has structures called transverse tubules that are extensions of the sarcolemma, the cell membrane, that extend deep down into the cell. So if you look at this image, so right here we've pulled a bit of the cell membrane back so that you can see inside the cell. These little green, very thin little lines you see right here, these are T-tubules. And I wish they had maintained the same color, because since it is just a part of the sarcolemma, it should be the same color as the sarcolemma. The next image is going to show you this again on the next slide, and hopefully it will illustrate it a little bit better. Now, because these transverse tubules are made of cell membrane, they have the same characteristics as the cell membrane, which is they have voltage-gated channels. All right, so if you look at this image here, you can see this entire structure, of course, is one muscle fiber, which is one muscle cell. Here you can see the cell membrane, which is called the sarcolemma. And I really like this image because it keeps everything, the color is consistent. So the cell membrane is this light bluish gray. Your transverse tubules are the light bluish gray that transverse or go into and through the cell. Okay, so you can see transverse tubules coming down from the cell membrane into the sarcoplasm of the cell. And here you can see it again. So these actually surround these myofibrils. So a skeletal muscle fiber is made up primarily of myofibrils. They extend the length of the muscle cell. Well, What's a myofibril? We're going to get there, I promise. Let's get some of this other stuff first. So myofibrils, remember, are going to make up the cell membrane. They're bundles of smaller little pieces called myofilaments. Boy, that's a lot of myo stuff, right? So these are myofilaments, okay? These little thin things you see in here, those are myofilaments. They make up a myofibril, and the myofibrils make up the muscle fiber. And then the muscle fibers all bundle together to make, yeah, come on, you know it, a fascicle. And then the fascicles all bundle together to make the muscle. Layers and layers of tubular structures all together, right? Okay, so we talked about the transverse tubules coming down from the cell membrane, still being a part of that cell membrane. Well, they're right there closely packaged in with the sarcoplasmic reticulum, which is a modified type of smooth endoplasmic reticulum, smooth ER. So if you look at this image, all of this net-like stuff that's running around those myofibrils and comes to these little enlarged areas right there beside your T-tubules, all of that is your sarcoplasmic reticulum, okay? So where you have these bulges of the sarcoplasmic reticulum right there on each side of your T-tubule, those are called terminal cisternae. So they're sort of like little sacs of the sarcoplasmic reticulum. Now, the SR, sarcoplasmic reticulum, is going to store calcium. We'll talk a little more about that. But this area right here, where the terminal cisternae and the transverse tubules come together. Now remember, you've got the terminal cisternae on both sides of the transverse tubule, so there's three parts there together. We call that a triad. Now your sarcoplasmic reticulum has calcium pumps. Your sarcoplasmic reticulum stores calcium. The calcium pumps will help to pump calcium in and out of the SR. The calcium that's stored in the sarcoplasmic reticulum is bound to calmodulin and calsequestrin. So think about that word, calsequestrin. What does it mean to sequester? To keep in place. So these bind to the calcium, keep them contained within the sarcoplasmic reticulum. But those calcium channels located in the sarcoplasmic reticulum allow the calcium to move out of the sarcoplasmic reticulum when that wave of depolarization, the electrical signal, travels down the T-tubule. It causes the SR to release calcium into the sarcoplasm of the muscle cell. Let's look at this microscopic anatomy some more now. First of all, let's go back up here and make sure we still know where we're at. So we're looking at a muscle cell, also called a muscle fiber, which is made up of bundles of myofibrils. That's these little tubular structures right here. But your myofibrils are made up of even smaller little myofilaments. Okay? Your myofilaments are one of two kinds. Your thick filaments are bundles of a protein called myosin. The myosin molecule has two heads that are toward the end of the filament. Okay? So all of these little stringy things right here are these all wound together. Okay? Are all bound together. So this is a molecule of myosin. This is a myosin filament. Each molecule has the two heads, and notice each head has a binding site for actin and, oh, there it is again, ATP. Yay! All right, guys, sorry, I get silly every now and then sitting here talking to my computer by myself. All right, so if there's an actin binding site on the myosin heads, that should tell you they want to bind to actin. So actin is the thin filament. Now look again, we're looking at these little things right here, the very teeny tiny little strings. Myosin is the thick filament with two heads. Myosin is a double strand of actin molecules. Now, to me, I think of it like a pearl necklace that's been twisted, and it creates that twisted helix with two strings of pearls. Now, in addition to those, so the little red pearls would be your actin. See the little black spots right there? That is a myosin binding site. So these myosin heads with their actin binding sites want to hook up with the actin that has myosin binding sites. So your actin has sites for myosin, your myosin has sites for actin, so they can hook up together. But now notice you've got that green rope-like thing that's covering all the binding sites on the actin. So look right here. This is called tropomyosin. It's covering the actin binding sites. And it's anchored in place by these little globular proteins called troponin. So here's how I think of it. The troponin is an anchor that holds the tropomyosin. Trop is a rope. It holds the rope over the binding sites. So as long as all of this is in place, poor old actin up here can't get with his girlfriend and hook up. Okay? Because the binding sites are covered. So troponin, the blue proteins, and the tropomyosin, the green rope, these are called regulatory proteins because they regulate when actin and myosin can get together. I know this video is getting a little long, but bear with me. We just have a little bit more on the microscopic anatomy that we've got to understand before we can talk about muscle contraction. Okay? So, what we're looking at here, again, you've got a muscle fiber, which is a muscle cell. Okay? You've got your myofibrils within, making up the muscle cell, or most of the muscle cell. Then you've got your tiny little myofilaments, your actin and myosin, making up the myofibril. Okay? So when we look at those structures with a little more detail, one of the most important things to recognize is the sarcomere. So a sarcomere, here you have three of them. 1, 2, and 3. A sarcomere runs from this to this. So that's 1. From here to here, that's 2. From here to here, that's 3. And you can go this way as well. So these little zigzag lines are called Z-disc. Okay? So that's the one that I want you to focus on. Now, the A band, the I band, the H zone, the M line, that's all important. But it's not necessarily the most important thing to focus on. So I want you to understand the sarcomere is delineated at both ends by the Z-disc. So that simply means from Z-disc to Z-disc is one sarcomere. So this appearance you see of being striped is simply the overlapping thick and thin filaments that we've seen already. Okay? So there are specialized proteins that run perpendicular to the myofilaments that serve as anchors for those actin filaments. Now, we're going to see those as well. Just hang on. Let's continue with this sarcomere. So when you look with more detail at this sarcomere, okay, here's a Z-disc, here's a Z-disc. So all of this is one sarcomere, okay? There's your thick filament, the myosin. There's your thin filament, the actin, okay? So far so good, huh? Nothing new. I mentioned a while ago that there were other proteins that anchor these filaments in place. Well, if you look right here at these green little coiled up stringy things, those are actually one of those proteins. It's called connectin, also called titin. So it's a twisted cable-like filament that attaches to the thick filaments and helps to maintain their alignment, but they're coiled up and sort of spring-like. So when the sarcomeres actually shorten, leading to muscle contraction, they get compressed and produce a bit of tension that's then released, and it allows the sarcomere to go back to its resting length. So think of it sort of like a spring. You pull it and then you let it go back. So when you pull on it, it pulls that myosin, holds it in place, and then relax. It allows the myosin to go back. All right, so let's wrap up this microscopic anatomy by looking at a few other things that are found within muscle cells. Now, obviously we know that cells have mitochondria that we use to create ATP. Muscle fibers have lots of mitochondria because they are constantly using ATP. Therefore, we must restore those ATPs by creating more. Myoglobin is found within a cell that is a molecule that actually stores oxygen. So there is a ready supply of oxygen to use to produce that ATP. Myoglobin is a pigment molecule that has sort of a reddish color, and it's very similar to hemoglobin. But when you have muscle tissue that has a lot of myoglobin, that muscle tissue appears much more dark. So think about chicken. Dark meat chicken, more myoglobin. We'll talk more about that when we look at energy usage by these cells. So a single muscle fiber can have somewhere around 300 mitochondria. So we're going to be making a whole lot of ATP there, huh? Yeah. So we also store glycogen, because remember, glycogen is made up of individual glucose molecules all hooked together. So those mitochondria have plenty of sugar ready to use to make ATP. In addition to that, we have something called creatine phosphate. Phosphate, what's the importance of that? We can steal that phosphate. And when we steal that phosphate, all we have to do is hook it onto an ADP, and then we've made more, ATP. All right, let's wrap up all this microscopic anatomy by looking at a disorder associated with a protein called dystrophin. So dystrophin is part of a protein complex that anchors myofibrils that are right next to the sarcolemma to proteins that are within the sarcolemma. So the dystrophin links internal myofilament proteins of a muscle fiber to external proteins. Okay, so that's what this is when you hear me talk about dystrophin, okay? So muscular dystrophy is a term that actually includes several different hereditary diseases where the skeletal muscles degenerate, lose strength, and then tend to be replaced with fibrous connective tissues and adipose tissue. The new connective tissues then can impede the blood circulation, which further exacerbates and accelerates the muscle degeneration. Duchenne muscular dystrophy, DMD, is the most common form. It is almost exclusively found in males. It occurs in about 1 in 3,500 live births, and it results from the expression of a sex-linked recessive allele, and that is the gene that helps dystrophin to be made. So if the gene is defective, you're making a defective product. So the dystrophin either has an abnormal structure, or it can be produced in just not enough of the product to be effective. So either it's bad or there's not enough. This results in causing the sarcolemma to be unstable. It's susceptible to damage by the forces that are generated by muscle contraction, and then the sarcomere is non-functioning because you've damaged the proteins. This allows excess calcium ions to enter the muscle fibers and damages those contractile proteins, with loss of muscle fiber accompanying that. So the muscular difficulties tend to become apparent during early childhood, and you see this usually for the first time when they're learning to walk. So the child will fall frequently or have difficulty standing up after falling. The hips tend to be affected first, followed by the lower limbs, and eventually the abdominal and vertebral muscles as well. This muscular atrophy causes shortening of the muscles, resulting in postural abnormalities such as scoliosis. I know you learned about scoliosis when you did the bones. Remember, that's an abnormal lateral curvature of the spine. Now, DMD is incurable. We can't fix it. So patients tend to be confined to a wheelchair by adolescence, and they rarely live beyond the age of 30 because death typically results from respiratory and heart complications, both which rely on muscle contraction.